So what influences cell division? I assume that you would think immediately of things like chemicals, nutrients, vitamins, and so on. And that's certainly one group, essential nutrients. But we also have to think about other things, including like growth factors. So when we talk about you growing, we talk about growth hormone. Growth factors stimulate division or increase the rate of division of cells. So that's how you grow. Density-dependent inhibition. If something is inhibited, it's slowed, right? Density-dependent. Well, this means, are you crowded? Cells don't like to be crowded. If they're crowded, they'll actually stop dividing. This is good because we don't want the cells to run out of nutrients. We don't want them to be crowded and uncomfortable. We don't want toxins to build up because they're too close together. So this is okay. Anchorage dependence. Well, if you're dependent on something, you need it, right? If you're anchored, what does that mean? Like a boat anchor fixed in one place? So this actually means that cells need to be in contact with some sort of solid surface before they're willing to divide. So if you add growth factor, you can get the cells to grow faster. And if you remove cells that have grown in a single layer, they will actually fill that space again. So there is a lot of evidence in biology to show that these factors are very, very important for maintaining happy, healthy cells. But we also have to look at the fact that the cell cycle needs to be under some sort of check and balance system. We can't just have the cell cycle go and go and go with no control. So we actually look at having checkpoints, and some people look at this like border crossing. Okay. Are you allowed to move to the next phase? This will actually cause an event to stop or allow it to go. So I like to think about it more like red light, green light. What events might this actually control? Well, we can look at control of growth. We can look at control of actual division. And we can actually look at control of whether or not the DNA will go through replication as well. So we have a lot of control possible points. The three major ones that we see in most eukaryotic cells are the G1, G2, and M checkpoints. Well, G1 is growth, right? So you can stop the cell cycle in the first growth pattern. You can also stop it in the second growth pattern. Or, what would M be? Mitosis. So you can stop it right before mitosis begins. When we look at this process, the G1 checkpoint says, okay, do we have enough nutrients, resources, and signal to move from G1 to S? If it's a red light, no. If it's a green light, yes. Then we've got another one. Do we have enough resources? Do we have enough signal to move from growth to mitosis? So you can actually find cells with a nucleus in its replicated state that's just sitting there because it got arrested or stopped before it went into mitosis. Then we can start mitosis and stop it partway through as well. So we will see some cells sometimes that are kind of in the state of they've started to do the whole telophase cytokinesis thing and kind of got stuck. This is a very good system for your body because we've got to regulate these cells. Without regulation, you get what we call uncontrolled cell division. Uncontrolled cell division is a fancy way to say cancer. When we look at the induction of cancers, we're finding that more and more of them are occurring because the cells have stopped listening to the cell cycle controls. So the cells divide rapidly. They don't care if they're crowded. And then they can actually start to spread to other tissues and places in your body where you would not normally expect to find them. So without inhibition or without checkpoints, cells can go into a cancer state or uncontrolled cell division fairly easily. Now, we do look at mutations in the cell cycle, and typically it would take at least 2 major mutations to have cells start into a cancer state like this, but it does occur. So the difference between cancer states or cancer tumor types is when we look at benign tumors, they're actually going to stay at the original site. Malignant tumors are the ones we really get worried about because they will what we call metastasizing, and they'll spread to other locations. Why are benign tumors better? Well, these are easier to treat typically. Typically involves surgery to remove the tumor itself and then chemo afterwards to help control any further cell division. Malignant tumors that have spread to the entire body can be very difficult to remove, and often we're into a full chemo radiation regimen, and it would be high dose chemo to help to try to control these or the spread. Cancers are named based on where they are actually found originally in the body. So carcinomas are associated with surfaces or body coverings. So skin, things like that. Sarcomas are actually associated with supportive and connective tissue. And then leukemias we associate with blood tissue types, either blood forming or blood itself.